Hello class. Today's topic on that subject cited biotechnology and its application uh, in the field of medicine. So today we have going to study the biotechnological applications in the field of medicines. Actually, the recombinant DNA technologies process have made immense impact in the area of healthcare. So the question is, what advantage is of the recombinant therapeutics? Number one is the enable mass production. Number two is the safe and more effective drugs we will find, and do not induce unwanted immunological response as is common in case of the similar products isolated from non-human sources. So, biotechnological applications in the field of medicines are the diagnose and treat different diseases with effective recombinant therapeutic drugs, protect from the dangerous diseases also. Through gene therapy, new and healthy genes can be inserted to replace the damaged cells. And the targeted action of drug by thorough study of the genomics. The recombinant therapeutics do not induce unwanted immunological response as in case of the similar product that is isolated from the non-human sources. At present, about 30 recombinant therapeutics have been approved globally, and 12 of these are presently being marked in India. In India, these products are human insulin, streptokinase, erythropoietin, hepatitis B vaccine, human growth hormone, human interleukin, granulocyte colony stimulating factor that is known as the GCSF, granulocyte macrophages CSF that is also known as the GM-CSF, alpha interferon, gamma interferon, blood factor 8, and follicle stimulating hormones. Some of the human proteins that have been synthesized from genes cloned in bacteria and or from the any eukaryotic cells, or by farming, all of these except hepatitis B are being imported and consumed in India. So a branch that is known as the biopharmaceuticals, uh, it is the medical drug produced branch by using the biotechnology. They include the protein or including the antibiotics formation, development of the nucleic acid, and production of the RNA and DNA, or we can say the antisense oligonucleotides also. Now, what are the genetically engineering insulin? We know that insulin synthesized by the beta cells of the isolate of Langerhans in the pancreas, and it controls the level of the glucose in the blood. As insulin deficiency manifests itself as diabetes mellitus, whose symptoms can be alleviated by a continuing program of the insulin injections, thereby supplementing the limited amount of the hormones that is synthesized by the patient's pancreas. Insulin used for the diabetes was earlier extracted from the pancreas of the slaughtered cattle and pigs, but animal insulin is generally satisfactory through cause the allergy in some cases in some patients. So deficiency of insulin that cause the disease which can be controlled by this is giving as a regular intervals. Traditionally we use this from the salted. So bacteria were now genetically engineered to produce the human insulin in a large quantity, as the bacteria can easily grow in the culture media. So first it is created in 1983. Eli Lilly is an American company which is going to be developed for the first genetically engineered insulins and insulin hormone to regulate the blood sugar level with the diabetic patients. The mature insulin have the two short polypeptide chain A and chain B, which are linked by a disulfide bridge. This insulin synthesized as a prohormone in all mammals, including human being. A peptide that B peptide chain, an additional C peptide chain absent in the mature insulin, as it get removed during the process of the maturation. So using the recombinant DNA technology, two DNA sequence corresponding chain A and B of human insulin introduced into the plasmid of E. coli bacteria produces the insulin chain separately. The chain A and B extracted and combined by the disulfide bond, that is the human insulin. Look at this picture in which a proinsulin is there. B cells peptides, when C cells is removed, C chain is removed, there is the free C peptide is free now, and we get the insulin. So actually in human being, this gene which is responsible for the production of this hormone, it is located on chromosome number 11. So we are going to prepare this chain A and B were produced separately and is extracted and combined by creating the disulfide bond to form the human insulin by using this bacterial cell. So this is the genetically engineering of insulin. In this picture you can see this diagram, the synthetic DNA encoding B chain and synthetic DNA encoding the A chain, red color. It is inserted in the bacterial cell to form these B polypeptide chain and A polypeptide chain, and then it is going to be purified and mix this chain with the sulfide bond, A chain polypeptide bonds. This is known as the human insulin that is prepared for the diabetic patients. Second thing is the gene therapy. In the case of the medical field, is a corrective therapy to correct or treat the hereditary diseases that a gene defect that is diagnosed in the early childhood age or in the embryo. The method where genes inserted into the cells and tissue to treat the disease. The basic principle of this gene therapy is that a normal gene, when get inserted into individual cell or embryo with gene defect, the normal cell will take over the function of the compensate for the non-functional gene. This first clinical gene therapy is given in the 1990 to a 4-year-old girl with the deficiency that is known as the ADA deficiency, adenosine deaminase deficiency. Initially it is treated by the bone marrow transplantation and by the enzyme replacement therapy. Now what method was used to treat the ADA through the gene therapy? The ADA deficiency disorder is a basically hereditary disorder that is caused due to the deletion of gene for the adenosine deaminase. This enzyme very crucial for human immune system function. So in gene therapy, this lymphocytes from blood of the patients are isolated and it is grown in a culture media. The functional ADA complementary DNA introduced into the lymphocytes through the retrovirus vector and returned to the patients. Since lymphocytes have short lifespan, we know it is a WBC, so very short lifespan, near about 1 week, and genetically engineered these lymphocytes are periodically introduced into the patients. But if gene isolate from the marrow cells that producing the ADA introduced into the cell in embryonic stage, so we will get the permanent cure of this problem. This is the gene therapy. Number 1, the cell harvest from the patient. Number 2, in lab, this with the use of this virus altered, so cannot reproduce. And in the third step, a gene is inserted into the virus altered virus mix with the patient's cell, and then cells becomes genetically altered, then altered cells injected into the patient's body again. This altered cell produce the desirable protein. So this is the treatment with the help of the gene therapy in the medical field for the human being. Third very useful diagnostic technology is known as the molecular diagnostics technique, that is used to diagnose and monitor the disease, detect the risk, and decide what happen or what therapies will work best for this individual patients. So treatment of a disease be effective earlier diagnosis, and understanding the pathophysiology is very important. So conventional method of diagnosis, that was the serum and urine analysis only, and early detections of disease were not possible by the serum and urine analysis. So early diagnosis possibility with the advanced molecular diagnostic like recombinant DNA technology, or we can see it is a RT-PCR, polymerase chain reaction, that is the enzyme linked immunosorbent assay, ELISA test, or these are the earlier diagnostic possible technologies. Conventional methods that detect the presence of pathogens, means that bacteria and virus, when disease is well established and produce the disease symptoms, at that time pathogens are very high in the human body. So significance or very importance of this molecular diagnostics is that molecular diagnostic like PCR detect the pathogens even at a very low concentration present in the human being, and it amplification of the nucleic acids of the pathogens, so we will get the earlier result. And detect the HIV, human immune deficiency virus, is suspect in the suspected AIDS patients, and detect the mutations also in the genes in suspected cancer patients also, what mutations are there, and detect this molecular diagnostic technique may detect the genetic disorder also that carries from generation to generation. The PCR requires a single standard DNA or RNA that is the tagged with the radioactive molecules to hybridize the complementary DNA in clones. And the following hybridization, the next step is the autoradiography also. So clone with mutation or mutated gene that not appear in the photographic film, the probe will not have the complementary with the mutated genes. So this is the significance of the molecular diagnostic techniques. Here is the principle of the PCR. Earlier you have learned in the previous chapter that the clinical sample is there, the DNA is preparation for that, and then amplified the target DNA and detection in the system, so we can detect the PCR. So millions of copies in the 30 cycles we will get. For that, a PCR device, a PCR machine, that is the thermocycler, is used for that based on the principle of the polymerase chain reaction. ELISA is another molecular diagnostic technique, very important diagnostic technique to detect the antigen or the foreign substance. What is the antigen? Antigen is the foreign substance or that developed or formed by the attacking of any kind of pathogen. So it is a foreign substance or pathogen. It is known as the antigen, and it works on the principle of antigen antibody reaction. Means the ELISA detect the pathogens by presence of the antigen. That antigen in form of the proteins, sometimes glycoprotein, sometimes other chemical molecules, or by the antibodies synthesized against the pathogens. So we will get know the what antibodies are synthesized against that infection of that particular pathogen. This is the ELISA technique, antigen antibodies binding technique. An antibody is a protein that produced by the our immune system, by the human immune system, in response to presence of any antigen. When there is any antigen is present, our antibody is produced from our immune system, and it is going to be bind that antigen. So ELISA is also based on this technique, the antigen antibody binding technologies. So This is all about the role of this biotechnology in the medicine field. Thank you and have a nice day.